and we go further to our next speaker, Dr. Gurevich. I think I was seeing him here. Dr. Gurevich, are you here?
Yes. Yes.
Okay.
I'm
okay,
here.
Great.
But do I understand correctly that Nikolausakis wanted to be?
Will be the last one.
Okay. So he wants to be the last one.
Yes.
Okay.
So now we go to our next speaker, Dr. Gurevich, who also represents a quite big group of scientists from Augsburg University, Denmark, and the Dmitry Mendeleev University of Chemical Technology of Russia. And the title of the presentation is How the Drug Loading Method affects cellular uptake of PVP-based nanocarriers. Can we have this share screen
yes.
with the presentation?
For me, can you see it?
Not yet.
Okay. I just need to press share.
Okay, now we can see, I think.
Okay. Right, thank you for introducing me. And yes, this is my talk, how the drug loading method affects cellular uptake of PVP-based nanocarriers. Right, so we did it together with our colleagues from Dmitry Mendeleev University. Right, okay. So, let me see, just a second. Right. Okay, so what is the material we work with? Can you hear me?
Yes.
Right. So that's PVP block copolymer synthesized by our colleagues at Mendeleev University. So it consists of a hydrophobic block, so which is just 17 carbons long. Can't get my mouse on the screen. And a hydrophilic block, which is PVP in this case. And the length of this block can be varied from 1 kilodalton up to 12 kilodalton. So in other words, it means that we have between 10 and 120 units, roughly, of the polymer. Okay. Yes. So why this system is nice and interesting? So because essentially we can create any drug delivery system based on such polymers, right? So in solution, the hydrophobic blocks will form a hydrophobic core, and the hydrophilic segment will form a hydrophilic corona around it, which can be quite large. So, and if, for instance, we're using several different polymers with different modifications, so we can have a micelle like shown on the screen, and this micelle can have, for instance, some targeting agents that can attack particular cells. It can have some contrast agents that can allow us to follow these micelles in the body by NMR, for instance. Okay, and of course, it can carry a drug, so a hydrophobic drug, which can be loaded into the core. From our perspective, it's very interesting to figure out whether we can create micelles like this, and whether this micelle will be similar to what we draw on the picture, to this ideal picture. Okay, that's one issue, and another issue is it's interesting how these micelles will be uptaken into individual cells, right? Okay, so that will be the subject of my talk. First of all, about structure of the micelles, right? We can deposit these micelles on, on a substrate, on atomically flat substrate, in this case mica, and just visualize. So that's AFM image. And you can see that, well, it looks like this schematic picture that we draw. So we see hydrophobic core, right? And we see a corona, which is obviously collapsed on this hydrophilic substrate. So it's in line with what we expect, right? So how can we load this micelles? So in the literature, many different technologies are known. So it can be a direct dissolution of the drug. It can be a solvent evaporation. So it can be made through oil and water emulsion, ultrasonic suspension, direct dialysis, and co-solvent evaporation. We started to work with ultrasonic suspension and oil and water emulsion. So that's the easiest technique. So, and I would say and both of them are very commonly used in literature, right? So if we compare, for instance, the results of ultrasonic suspension and oil and water emulsion, we see that generally speaking, ultrasonic suspension creates particles which are which can be significantly smaller. So that's the solid line shown here in the lowest graph. So, and we see that, well, we have a quite large fraction of particles that are smaller than, yes, 100 nanometers. So in the range about 30, 50 nanometer, right? So, and if we ask ourselves the question, what we actually expect from the picture, so, and we calculate size of our polymer, so then we will see that our micelles should probably have a size around 30, maybe max 50 nanometer. So no more. So everything that is larger than that are aggregates, right? So aggregates of micelles. As we see by doing oil and water emulsion, we mainly create aggregates. If we do ultrasonic suspension, so we create a large fraction of small particles, but also quite a bit of aggregates. So if we, for instance, filter out these aggregates with centrifugal filter, so then we will find a distribution pretty much similar to oil and water emulsion, right? The next question is, are the small particles, particles that are similar to real micelles, are important for drug delivery to cells, for cell uptake, right? So we perform a number of experiments involving the culture of glioblastoma and culture of fibroblasts. So glioblastoma is U87 here, and fibroblast is CRL2429. Okay, and what you can see here, okay, in this experiment, the model drug was curcumin, right? And curcumin is nice and safe material, but it's also a fluorophore, so we can, not so good fluorophore, but it is a fluorophore, and we can see it in fluorescent images. Right. So what we can notice here is that for emulsion prepared drugs, for emulsion prepared suspension, we see that our micelles penetrate into the cells. So, and right, yes, you see this yellow halo on composite images, and yeah, they are localized somewhere around nuclei, but they do not enter nuclei, right? Okay, if on contrary we prepare, say, suspension, ultrasonic suspension, so then we see that we don't observe any more this very well-defined distribution, but our model drug, our curcumin, seems to be distributed all around the cell. Right, so that's quite an interesting phenomena, right? Which obviously indicates that, say, the preparation method affects quite a bit the uptake of micelles in the cells, right? We can prove it by, for instance, removing the small fraction from from the ultrasonically prepared suspension and treat cells with this fraction without small particles. And then we reproduce exactly the same result as we got with oil and water emulsion. So we get a pretty sharp distribution of our, say, drug. Yes, we see that it is clearly localized in some cellular structure around nuclei, but it's, yeah, but we don't get the picture when it's distributed all throughout the cell. Yes. So that's then the question we can ask. So it's definitely small particles that are important for the purpose. And definitely small particles can penetrate into cells differently, but is it just the issue of their size, or it is related to something else? So for instance, we can maybe suspect that particles prepared by ultrasonication are not well formed and maybe unstable, and maybe they exhibit somewhat different uptake mechanism, right? To prove this, we decided to look a little bit more carefully at the morphology of particles, right? And here are some examples of what we can see. That's, for instance, STEM images of different particles before and after filtering. So if we plot distribution, so we can see that indeed filtration removes small particles. So that's this blue peak that you see. Now it's not covered by brown, so it means it is removed after filtration. And so it means indeed we reduce smaller particles in this way. As I said, we had different types of preparation. So we excluded 1 kilodalton aggregates because they made particles of irregular shape and focus on, say, larger polymer. So in particular, 12 kilodalton. So, but you see that 6 and 3 also work fine. And we decided to try a different method, which is co-solvent evaporation method. The reason why we went for this is that the DLS and NTA study performed on just polymer solution indicated to us that we don't get fully uniform polymeric solution, even if we dilute our polymer quite a lot. So we came to conclusion that water apparently is not a good solvent for the PVP-OD. So it is a good solvent for PVP, but for PVP-OD, so the PVP with this extra hydrophobic group, it's not a good solvent anymore. And we attempted to find a good solvent for this polymer, and we found that ethanol apparently works really very well. Okay, and we can create a good solution of PVP-OD without any aggregates. So at low concentrations, of course, right? So what we did then, we followed co-solvent evaporation idea. So we mixed large amount of ethanol with small amount of water, dissolved our PVP-OD in this ethanolic solution, and then we slowly evaporated ethanol. So it means we increase the concentration of water slowly and let our polymer molecules to slowly form the micelles, and also to slowly catch the drug into the core of these micelles, in a hope that in this way we will get much better defined micelles and much less aggregates. This indeed worked, right? So you can see a new DLS size distribution and also TEM size distribution. And now, as you can see, most of the micelles, most of the particles that we observe are in the range between 20 and 30 nanometer, right? And we have incredibly small amount of aggregates, right? Okay. Furthermore, we found that we can stabilize this solution of particles quite a bit by changing pH above pH 9. So we decided to make pH 10. And this, as we know, will create some negative charge on the particles and will lead to a more stable solution. Right. Okay, so this shows stability of our preparation, co-solvent evaporation prepared preparation of micelles. Over time, so after 3 weeks, you see the peak is still around, and we can also dissolve it in culture medium, so in this case, DMAM, and it is also stable, right? Okay, so here we wanted to see what will happen with this small preparation from, say, 2 different perspectives. One thing we wanted to see whether it will again demonstrate the same type of behavior as ultrasonically prepared suspension, so whether it will, say, penetrate indiscriminately through all cells compartment or not, right? And we wanted to see kinetics, and on the other hand, we wanted to see how we can block the penetration. So in other words, we wanted to see if endocytosis is the main mechanism for uptake of such particles or not. Right. Okay, so what do we see here? We see on the top line, time dependence of the penetration. Okay, initially, as you see, the exposure is a bit high, a bit long, so because we don't have much signal, but we see that our particles are absorbed on the surface. Initially, surface just shines in the first 10 minutes, and then after 30 minutes, the particles will start to penetrate in the cell. And after 2 hours, we see that the cell lights up completely, but the position of the particles is very well defined, right? If we do slicing of our cell, we see that the drug is localized around the nuclei, but doesn't penetrate into the nuclei. So nuclei are free of the particles. Now, if we, and most probably, they are located in lysosomes, right? We see a very good co-localization if we apply lyso tracker dye. Okay, now what will happen if we will try to block functioning of the cell? So will our micelles still penetrate into the cell or not? Okay. That's what you see in the lower line. So control is just uptake of our micelles at normal condition. Okay, now we can apply a dinosaur, for instance. So dinosaur will block dynamine-dependent uptake, right? And we see that dinosaur is actually does this, okay, but dinosaur has a little problem. As we found out, dinosaur is can bleach DII, so our model drug and fluorophore. So we decided to continue and see other possible inhibitors of penetration and see how they work. Sodium nitrite. Sodium azide. Sodium azide is essentially a toxic chemical that can block ATP in the cells, and you see that it slows down the uptake, but sodium azide is a bit tricky. It is used quite a bit in literature, but the problem is that if you see the concentration of sodium azide different groups use, it varies quite a lot. And the problem there is that if you add too much of sodium azide, you will just kill your cells. So if you will add too little of sodium azide, you will not block all ATP, and uptake is still possible. So we don't have much trust to sodium azide, but consider it as indication. What we found works the best is just cooling cells to 4 degrees C. So this slows endocytosis a lot, and we can see what happens. In this case, you see that the uptake of our micelles is very, very small, very minor. Okay, now let's do flow cytometry and compare what happens if we apply our nice and very monodispersed micelles obtained by co-solvent evaporation and micelles obtained by sonication. Right. So what you see here, you see a number of counts. So that's the number of cells on the vertical axis, and on the horizontal axis, we see the fluorescent intensity. So this will tells us to what extent the cells have, say, absorbed our model drug, our fluorophore. Okay. Right. What you see that say sonicated nanocarriers, so produced by ultrasonication, exhibit the highest intensity, so around 10 to 6 at 37 degrees. It means cells absorb a lot of them, right? So, and the intensity we get from co-solvent produced nanocarriers at 37 degrees, so that's violet, is actually an order of magnitude less than that. So interestingly, if we cool down now to 4 degrees, then we see that, well, the both of them are reduced, both intensities are reduced, but intensity for co-solvent produced nanocarriers is reduced significantly more, so almost by 2 orders of magnitude, 1.5 orders of magnitude, compared to sonicated nanocarriers. So that's an interesting phenomenon. So it means that when we cool down our cells, we block uptake of co-solvent evaporation produced nanocarriers, but we do not block uptake of nanocarriers produced by sonification. So what is the issue here? We can figure out this by doing fluorescent microscopy on the cells, and we see that nanocarriers produced by sonification actually intensively adsorb on the cell membrane, while co-evaporation produced nanocarriers do not adsorb. So, right, that's what you can see on the image on the right-hand side. Okay, so this indicates to us that it's not only the question of size that affects cell uptake, so the production method can affect it as well. So maybe we speculate that the cells that micelles produced by sonication are not complete and more reactive, and can therefore be easier adsorbed into the cell membrane and can be easily incorporated into the cell membrane, and possibly we can have like different mechanisms of the uptake. So, and one of them can involve, for instance, micelles releasing their load, releasing their hydrophobic drug into the membrane, and then the drug is distributed in the membrane, and during endocytosis process, so the pieces of the membrane will be essentially entering the cells, and in this way the drug can travel inside the cells without being encapsulated in the micelle, say the nanocarrier we produce. Right. Okay, so that's the conclusions, and I would like to thank you for your attention. Please ask questions.
Thank you very much. It was very interesting. If the people have questions, or I can ask some. If nobody, okay. Can you please? Maybe I didn't catch correctly. When you were talking about these two methods, ultrasound and solubilization, you showed the particles, and as I could see from that slide, the shape of the particles were quite different. But after, no, before this, but after some chromatography or centrifugation, it become like spheric size, spheric shape. Why? Because what I understand that ultrasound gives, yes, this one, ultrasound gives some different type of particles, and morphology is quite different. And maybe not all of them are micelles.
Yes.
Yes.
Precisely. That's what we also think. So that so in sonication, essentially the size distribution of your aggregates is produced due to sonication itself. So it's essentially you start with aggregates, and then these aggregates are broken down by sound waves, but they are not broken down to micelles as we draw micelles in textbooks.
But after centrifugation, why they become spheric again?
That's actually a good question. I don't have answer to this, because in principle, right, we centrifuge them on filter, but this is a fraction. The fraction that is shown here is the fraction that stayed on top.
Yes.
So if it would be a fraction that goes through, I can say, okay, it was some shear forces that.
Yeah, but I see that you have 30 kilodalton, so it definitely stays on top. Yeah.
Yeah. So that's the fraction that stays on top. Yeah. It is not clear why the particles became, say, smoother. Maybe it was just maybe there were still some shear forces. So when it was essentially shaking in the flask or something like this.
Okay.
Right.
Thank you.
Just time.
Thank you. If there are other questions, please please ask if any other questions. In audience, you can switch on the microphone and ask question now. Okay. Thank you very much.
Thank you.